Hi, my name is Kathy and welcome back. In today's tutorial, we're diving into the npm4 repo to explore more AI-generated protein sequences. These aren't just any proteins, they come with remarkable structure, sequence, and functionality indicators. Ready to follow along? Let's get started. The npm4 repo contains over 1,000 AI-generated proteins, all crafted from simple language prompts. Today, we're zeroing in on kinases. These biochemical powerhouses are responsible for phosphorylation, a critical process in countless biological systems. How do they make a real-world impact? Number 1. Cancer targets. Drugs like Gleevec target the BCR-ABL kinase, which is involved in chronic myeloid leukemia. By blocking this kinase, Gleevec effectively stops cancer cells from multiplying. Number 2. Diabetes targets. The drug metformin affects AMP-activated protein kinase, or AMPK, a kinase that plays a role in cellular energy balance. By activating AMPK, metformin helps lower blood sugar levels, making it a cornerstone treatment in type 2 diabetes. Number 3. Biotech breakthroughs. The New England Biolab's T4 polynucleotide kinase is widely used in research to add phosphate groups to DNA or RNA ends. This process is crucial for techniques like sequencing or creating probes used in diagnostics. This kinase enables advancements in personalized medicine and genetic engineering. Also, based on the analysis of the SwissProt database, about half of human proteome is actually poorly understood, and about 1/3 of the 535 known human kinases fall into this category. Designing and studying the so-called dark proteins could reveal new biological insights and lead to more precise, effective treatments. We filtered the npm4 repo and found 40 potential kinase candidates. Let's take a closer look at one of them. To do that, hover your mouse over it and click on More Details. We'll dive into the summary page where you will find the prompt used to generate it, the amino acid sequence of the protein, its prot-NLM predicted function, and the ESM-fold 3D structure prediction, which you can download via the little download button. Notice the F target? That's our protein's closest natural structural match, specifically PDB ID 5DT3. We can take this further into the 310 Copilot using the Edit This Button in Copilot button. On Copilot, we'll load this kinase directly from the repo. Since this is a kinase, let's ask Copilot to find and load an ATP molecule. ATP is a natural binding partner for kinases, and it's used as an energy source to power the kinase. Here is what the structure of ATP looks like. Now let's ask Copilot to dock them together, using diffdock under the hood. The result? A predicted kinase ATP complex that we can analyze further. Remember the F target result from the summary page? That's the closest PDB structure match to this AI protein, and that PDB ID is 5DT3. Let's compare the AI protein with 5DT3. I already have the PDB structure downloaded on my computer, so I'm going to upload it here. Now let's ask Copilot to compare the two complexes. Copilot runs two programs under the hood, TM-Align for the structural alignment, and then prot-NLM for the function prediction. Here we see the TM score is more than 0.9, which means the two structures are almost identical. Looking at the aligned structures, you will see that the ATP docked in the same pocket, which is great. Now let's compare some key kinase-specific features. Two of the important ones are called the HRD and the DFG motifs, which are highly conserved residues in most kinases. To have a clear view, let's reduce the cartoon representation to only the regions of the two motifs. We will visualize the cartoon of 5DT3 as green and that of the AI protein as orange. We can see that the motif regions from both structures superimpose very well. Now let's highlight the specific residues of the HRD and the DFG motifs as ball and stick, colored blue for 5DT3 and red for the AI protein. We can see that the highlighted residues of the motifs align just right. The DFG and HRD motifs surround the activation loop and are an important part of kinase activation and catalysis. This analysis suggests that the AI protein is a potential bona fide kinase. In conclusion, This isn't just a random sequence, it's a promising new kinase with indications of catalytic functionality that was crafted by a molecule programming AI. We hope this has inspired you to work with AI proteins too. Please visit 310.ai/npm/repo to explore, or go straight to 310.ai/copilot to get started. Thanks for watching, and see you in the next one.